Hello, and welcome back to Honors Bio. So today we're going to be talking about meiosis and how that can lead to genetic variation amongst the species. So we learned about the basics of the cell cycle and mitosis. Mitosis is how all your somatic cells replicate or duplicate. Basically how they go from one to two cells. Meiosis is how your sex cells, sperm and egg cells, respectively, replicate, how they reproduce. So that process can lead to differences in genetic variation. So let's get into it. So the first thing with reproduction is there are some organisms out there that do reproduce asexually. So because of that, they don't have specialized cells. They do not have sperm or egg cells. So they don't go through their bodies don't go through meiosis. So if it's a single cell organism, like most bacteria, they're going to reproduce asexually, which is just a splitting, budding. The fancy word is parthenogenesis, which is just, basically we take one cell and we end up with two. That's how they reproduce. There are a couple multicellular organisms that can reproduce asexually. It's very rare though. Those are the definition of clones. So the clones, what they are is they're genetically identical to the parent. Okay, so that's really all you need to know about asexual reproduction. Is that the offspring of asexual reproduction are genetically identical. So in sexual reproduction, so the rest of the multicellular organisms reproduce asexually, which is the fusion of two gametes to produce a thing called zygotes. Usually, it's during between a sperm and an egg cell that we get a zygote. What sexual reproduction does, this fusion of the zygote, it allows for greater genetic variation, which is beneficial to the survival of a species. And when we get to natural selection, you'll start to see why genetic variation is very critical to a species adapting and surviving. And then zygote will typically come from two different parents. There are a couple plants out there that are self-fertilizing. When we get to Mendelian genetics, which is our next unit with the Punnett squares, Mendel got very lucky because his pea plants that he was working with were self-fertilizing. So he could actually control traits and how they were passed down, which was very lucky. If he didn't done that, we probably would not be where we are at with predicting traits. So let's talk about chromosomes now. So we talked about homologous chromosomes in mitosis, but here they are again. And I want to stress this. Homologous chromosomes, the homologous pairs, okay, they carry the same gene. So let's say these homologous pairs, okay? This is chromosome 1. They carry the gene for eye color, but each pair or each homolog may have a different version of that trait, or we call that an allele, right? So they both may have eye color, but this homolog may have brown eye color. This homolog may have blue eye color. This homolog may have, short hair. Sorry. This homolog may have brown hair. That's a better example. This homolog may have blonde hair. That's right. So the homologous pairs, they have the same gene, eye color, bone structure, whatever it is. They just have different versions or different alleles of that gene. The exception is the sex chromosomes. Either the X and the Y chromosomes don't exist as a homologous pair. So the other chromosomes are known as autosomes chromosomes, and they all have homologs. So let's look at some pictures of chromosomes. So this is a picture of the chromosomes in a cell all lined up nicely together, so you can see the pairs that happen here. So chromosomes 1 through 22 are your autosomes. So all of these are your autosomal chromosomes. And then here is your X and your Y chromosomes, or your sex chromosomes. This, we call this a karyotype. This picture, this kind of chromosomal picture, has two X chromosomes indicating a female. So this is a human female, because we know that. One, because they tell us. Two, because they have 23 pairs of chromosomes with two X's. That indicates a human female. Oh, oops. This is what the human male chromosome looks like. So 1 through 22 still look the same, right? These two pictures look very similar when you look at the autosomal chromosomes. It's just the sex chromosomes that differ here. Males will have an X and a Y chromosome. So you can see the difference here for the X and the Y. The X is typically bigger than the Y in terms of genes too. The X chromosome carries like several thousand genes. The Y chromosome only carries like 100 genes or so. So let's get into meiosis now. So now they kind of have an idea of chromosomes and the homologous pairs, that they do carry the same gene, just different versions of that gene. Let's get into meiosis, because that's a critical part of understanding what happens in meiosis. So in humans, okay, right, we have 46 total chromosomes, or 23 pairs. This is shown here, right? We have 23 pairs, 23 times 2, because each of number has 2, is 46. Half of your chromosomes come from mom, the other half come from dad. Okay? So that means then that the gametes, the sperm and the egg cells, carry 23 chromosomes each. 22 autosomal chromosomes and 1 sex chromosome. That's that haploid number. The sex cells are haploid. Sperm and egg cells are haploid. All right? When fertilization results, when the sperm and the egg merge to form the zygote, the two haploids join together, they form a diploid. They form a diploid cell, 2N. All right? So make sure you know what cells are diploid and haploid. Okay? And again, the somatic cells, every other cells in our bodies reproduced by mitosis, is only the gametes, only the sperm and egg cells that reproduce by meiosis. So let's talk about meiosis in that process. Meiosis, I like to think of meiosis as mitosis happening twice. Because that's what's happening. All right? There's meiosis 1 and meiosis 2, and they use the same words as in mitosis: prophase, metaphase, anaphase, telophase. It goes through the same process, just twice. There are some differences that do lead to the genetic combination here. So the difference is that meiosis reduces the number of chromosomes by half. So in the end, so with mitosis, in the end, we got 2 cells, each with 46 chromosomes. In meiosis, at the end, we get 4 cells with 23 chromosomes each. So the daughter cells do differ from the parent. The 4 haploid cells at the end of meiosis are genetically different than the parent cell, where in mitosis the 2 daughter cells are genetically identical to the parent. Meiosis involves 2 divisions, hence meiosis 1 and meiosis 2. In meiosis 1, the first kind of go around of this, it involves a couple kind of special things. It involves a synapsis. So that's where the homologous chromosomes, that's where they'll pair up to form the homologous pair. We call it a tetrad. And then really, and you will see where the homologous pair, the 2 chromosomes kind of overlap each other. Where they overlap, we call that the chrismanta, because crossing over can occur where they're swapping genes. And we'll talk about how that process works. All right. In metaphase 1, the homologous pairs line up at the plate and in the middle of the cell. In anaphase 1, the sister chromatids do not separate, just the homologous pairs. And then overall, it's the separation of homologous pairs of chromosomes rather than the sister chromatids. So let's talk about meiosis 1. So meiosis 1, again, it's the same process as mitosis. We still go through prophase, metaphase, anaphase, telophase. We just call them, we just put the 1 in front of them because we go through it twice. So prophase 1, it's the same. The nuclear envelope disintegrates, the centrioles start moving to the poles, the spindle fibers start to form. Okay, but with the chromosomes, the homologous pairs line up to form a tetrad, and crossing over can occur during prophase 1. And then metaphase 1, it's the same. The homologous pairs will line up in the middle of the cell. Anaphase 1, spindle fibers will attach to the centromeres, pull apart the homologous chromosomes. Telophase and cytokinesis 1 still occur, where then we split and we get 2 daughter cells. And then we go through meiosis 2. So this is what meiosis 1 looks like. Okay. Interphase is still the same as it was with mitosis. With prophase 1, though, this is that tetrad. So this is that homologous pair that I was talking about, right? So the homologous pair line up with each homolog here. And then where they overlap right here, this is the chrysemma, okay? So when we talk about crossing over occurring between non-sister chromatids, that's what we're talking about. This crossing over really occurs between the homologs, between each kind of pair of chromosomes. So this is that tetrad. So really, metaphase 1 and anaphase 1 is just about separating the tetrad. It's just about breaking apart this. That's all meiosis 1 is just separating the homologous chromosomes. That's it. Meiosis, so then after we go through all of that, okay, meiosis 1, meiosis 2 occurs. And it's the same process. We still go through prophase, metaphase, anaphase, telophase, we just put a 2 next to it. Prophase 2, metaphase 2, anaphase 2, telophase 2. It's the same process, right? Prophase 2, centrioles, again, in each cell, move to the poles. Metaphase, chromosomes will lie at the plates. The spindle fibers attached to the centromere. In anaphase 2 now, this is where the sister chromatids get pulled apart. And then in telophase 2, right, we're done. Great, we get 4 haploid cells. This is what that looks like, okay? Right, so this is the end of meiosis 1, okay? So we get our two daughter cells to occur here. And then it just runs through the process again. All right, where prophase 2, where the centrioles are moving away, metaphase 2, the chromosomes line up in the middle of the plate, the spindle fibers attach, the sister chromatids, they separate, and then telophase 2 and cytokinesis occur to form the 4 haploid daughter cells. So that's why I think about it's meiosis is just mitosis happening twice. That's how I think about it, these stepwise, okay? It's still the same sequence of events. Prophase, metaphase, anaphase, telophase, cytokinesis is just our end products different. We get four haploid cells instead of 2 diploid cells. And then we get that genetic recombination of the crossing over and whatnot, which I will make a separate video to talk about the crossing over. So watch that video. Other than that, if you have questions, let me know. And I look forward to seeing you all next time in class.